Hello, and welcome to demola. And today's video, we're doing SNAB Biology 2024 Paper 3. Definitely the hardest of the lot. However, we're going to break it down, go through the questions, including the scientific article at the end, how to kind of analyze that and put the answers on the paper. So there's time to waste. Let's get into it.
Okay, so question one, it said one of the first plants to colonize some habitats is the fell cress. And it's saying that plant hormone IAA is produced in the growing tips of plants. So initially I'm kind of thinking, what might they ask me for? I imagine they're going to ask me about phototropism, surely. And then they might even ask me about succession, just because they've mentioned about colonization. But the first question we have here is state what term is meant by colonization of a habitat. And this one is a nice question to start off with. I think we should kind of know what colonization is, not even on a biological perspective. So yeah, just when a species moves into a new area. Okay, so next they're saying one role of auxin is to stimulate the phototropic response in shoots. So like I predicted, we've got a phototropism question. And then it says just explain how IA causes phototropic response. So this one should be fine. If you've learned it, you should be able to kind of just reproduce it. Apologies. If you learned it, you should just be able to reproduce it. So this is again something I'd recommend just learning. In terms of you're saying auxins accumulates on the shaded side. Very important that you say on the shaded side. And then what we're also going to say, this is a kind of an additional point that takes it higher than a GCSE answer. We were talking about auxins they kind of regulate how transcription factors work, and they promote the synthesis of proteins for cell elongation. So I think a lot of people might jump from accumulates on the shaded side and then it causes cell elongation. So some people might jump for that and you'll still get your mark for doing that. But I think it's just good practice to add in this middle bit about the regulation of transcription factors. And then finally, what you're saying, okay, if we're causing cell elongation from the shaded side, that means that we're going to bend towards the light. Yeah. Hopefully that makes sense. Then we've got here, so we said auxins stimulate the growth of lateral roots, which you can see on the diagram here. And then we're saying one receptor for IA is coded by the TIR1 gene. So scientists investigated the effects of auxin, plant homozygous for the genes, and heterozygous. So this is important. So we're thinking about study design. So I'm saying that we've got homozygous for both, and different concentrations of auxins are used. And then they've measured the mean number of lateral roots produced as like a sign of growth. So you have the data there, but let's quickly read the question. It says, explain why scientists used homozygous plants for this investigation. Now, the thing is, in that question, it's an explain question, so we need a biological basis. However, they're actually not telling you to refer to the data. So let's just ignore the data for now and just explain what's happening. So in terms of study design, why would you do that as a control kind of thing? Well, it's mainly because if we had heterozygous plants, so say we didn't do this, heterozygous would have both the normal and the mutated alleles, right? And the reason we don't want that is just to make sure, okay, the response we're getting is purely down to having the mutated allele. It's purely down to having the normal alleles. We don't want to kind of lead up to chance. I think on the mark scheme, it also mentions about like recessive might be hidden by the dominant and also codominance. I don't really think that's too relevant, and it is only a 2 marker. So saying these 2 points are going to be. Okay, so the next prompt is calculate the percentage decrease in the mean number of lateral roots for the fell crest with the mutant gene compared to the normal. So what we just want to quickly say here is, okay, this is the mutant gene, this is the new, and this is the original. So that helps us with formula because it's new minus old divided by old. Yeah, when you're doing a percentage change. So let's quickly just see here. So in terms of the normal one, I've got that at 1.4. If we just do it roughly. And then here we've got this at 0.8. So these are our values. So we said that new 0.8 minus old 1.4 divided by the old multiplied by 100 to get our answer. So you're going to get the answer as like minus 42.3% or something. No, sorry, minus 42.9% or something. So I'm just going to do 42.9 because it said calculate percentage decrease anyway. Okay. Now it says deduce the effect of the mutation in the TIR1 gene on the response to the fell crest to auxin. Now, if we're saying deduce, that means we need to use information or use data to come to a conclusion. So if we just look at the data, what you can see here is that the normal one is literally always higher. So at every single concentration of auxin, our normal is higher than the mutant gene. So I can just say mutation causes a decrease in response to IAA. Yeah, cool. It's only a 2 marker, so I'm not too bogged with doing a calculation. But what we can say is at every single concentration, yeah, the lateral roots produced with the mutation is lower. Yeah, and that should be fine to get you 2 marks. I think if you now want to go the extra step, you could maybe give a reason why. I don't think you necessarily need to for a deduced question because it's just kind of coming up with a conclusion based on data. But if you really wanted to add it on, you can say that the change in the shape of the receptor is caused by the mutation. And in that case, that means auxin can no longer bind to it. But that's kind of an additional point, which is nice to add. But in terms of a deduced question, it's the first two that. Because the question 2, if we read the context, so pathogenic bacteria are resistant to, can become resistant to antibiotics. And you've got the case of MRSA, which is resistant to many antibiotics. So we've got a chemical isolated from a licorice plant, and we're talking about antimicrobial properties with this practical that you should have done. So just in terms of what the first part is asking us to do, we can see we need to calculate the diameter and find the area. So just making sure we're going to the right one, we're going to the 80 micrograms. And we need to measure the diameter across here. So what you'll see is that's 19 millimeters. And then with that, what we're going to do is 19 is the diameter, so we'll put 19 in the table. I'd put 0.0 just because we've got a decimal point. Divided by 2, we get 9.5. And then we just need to do pi r squared. So pi multiplied by 9.5 squared. When you round your answer, you're going to get 284. Okay, so something that when you were doing this practical should have been really emphasized to you was the importance of aseptic technique. And I think aseptic technique is really important in terms of just memorizing the different things that you kind of have to do for A-level biology. These are some of them. So making sure you're washing hands and wearing gloves, making sure that you autoclave or flame the forceps to sterilize. Also, when you're kind of doing the practical, only lifting the lid slightly because it's going to prevent the entry of air contaminants, and also making sure you clean the tables before and after. So those are just things to mention. And really, when they're talking about describe how like the filter disc containing the antimicrobial can be safely placed in a bacterial lawn, literally they do just refer to making sure that you're using aseptic technique. That's what they really want to see from you. Okay. Now, part C, explain how an anomalous result could be identified. This again, the main thing is going to be plotting a graph. So if you plot a graph of like the different sorts of the different concentrations that you've used, and then also the amount of inhibition that happened, yeah, you can draw a line of best fit, and then any anomaly you would just see because it doesn't fit the trend. I think another thing you could do is maybe calculate like means or standard deviations or something, and any result that's outside of that standard deviation you would say is an anomaly. That's something else that's in the mark scheme, but I think this one is really the one that you'd be more expected to say. Okay, so we said photosynthesis is taking place in your chloroplast, and we said the light-dependent reaction can be investigated using the Hill reaction. Cool. So we know what we're talking about. So we've got this graph, we've got different herbicides. I'm just looking to kind of see if I can pick something out initially from the method. But then with isolated chloroplast, I'll just mention that as well. And we're looking at the Hill reaction. Okay. So location of the light-dependent reaction, this is just something that should be just known is thylakoid membrane. That's just knowledge to know. In terms of calculating the gradient of the slope for herbicide A at 50% inhibition, let's see. So normally, to be honest, I would say calculate a gradient at 50. So we've gone to 50 and we've gone across. Now, normally I'd say calculate the gradient. However, this is actually quite straight. So if you try to draw like a gradient here, you'll see that it's actually quite straight, to be honest. But I think it's good practice to draw the gradient anyway, regardless. You can see here, we're going down by 16, so you need to make sure you know. So if there's a difference of 20 between what is 10 spaces, then you know each space is worth 2. So that's how we can say the height is 16, and the length, we can see here, okay, there's 10 spaces between 2, so each space is 0.2. So that's how we get that 0.6. And then we can do 16 divided by 0.6, which is going to give us 26.67. But you also have to look significant figures. So we're going to leave that as an answer of 27. Now, describe the role of chlorophyll in the light-dependent reaction. This is something that we should know. So I always say define first. I think describe questions tend not to be too bad. But just quickly, definition. So I say chlorophyll is a photopigment. Yeah? What do photopigments do? Absorb light. So then we need to now use our knowledge of the process of the light-dependent reaction. So what happens when we absorb light? The electrons get excited, right? And when they get excited, they're going to travel towards the electron transport chain. I'm happy to kind of leave my answer at that because anything beyond that, I don't necessarily think is the role of chlorophyll. So that's how I'd leave. This is another describe question. So again, if you've done the learning, you'll know how to answer the question. So again, definition is what you'd always start with. So Rubisco is an enzyme. Cool. What do enzymes do? They catalyze reactions. This one specifically in the carbon cycle catalyzes the reaction between RuBP and carbon dioxide. If you want to say carbon fixation, that's fine. Whatever you say, just know you're relating RuBP to CO2. Cool. Now, what does that form? Well, you can say it forms an unstable 6-carbon compound intermediate, which breaks down into 2 GP molecules. But I've kind of just hopped the gun because it's only a 3 marker. So I've said it just forms 2 GP molecules. But you can say unstable 6-carbon intermediate, which breaks down into 2 GP as well. I don't think that's a necessity to go further in the carbon cycle. Okay, so we're going to have to do a comparison between 2 of these marine animals. Okay, so it's saying that the typical adult great white shark has a mass of 600 kilograms, whilst the humpback whale has a mass of approximately 140,000 kilograms. Cool. So we can see the trophic levels here. Immediately what I can say is, okay, well, humpback whale is a secondary consumer, whilst the great white shark is a tertiary consumer, which means that there's more trophic levels for the great white shark. Cool. So now we're saying, explain why an area of ocean can support a greater biomass. So it's going to somehow relate to this, we're thinking. And what we should know is that energy is lost at each level in the food chain. Yeah? So we come back. Now we're saying, okay, the community the humpback whale is in has fewer trophic levels. Cool. Energy is lost at each trophic level. That's something that we should know. Cool. So if there is going to be energy lost at each trophic level and there's less of them, then this means to me that a humpback whale is going to have more energy available to it, which it can then store as biomass, because we have to link back to the question in which it said that there's greater biomass. We need to link it back to that, which is why we're going to say that. Okay, so we've said that phytoplankton are photosynthetic unicellular microorganisms, and they are a primary producer. So first of all, state these questions normally just assess your knowledge. So you should know GPP equals MPP plus R, MPP is GPP minus R. Fine. Then we're saying concentration of oxygen in water can be measured using an oxygen probe. So explain how the oxygen probe can be used to estimate the effect of temperature on GPP. So initially, first one I'm thinking, okay, GPP equals MPP plus R, but we can't actually measure GPP by itself. So we need to measure MPP and R. Cool. Practical question, always, just always, please, do variables to start off with. So independent variable, and I think really for a 4 marker especially, it's a good approach to have. So independent variable, I'm thinking, okay, it's going to be the temperature, because that's what we're measuring, right? So, and also you can maybe highlight that, the effect of temperature. Cool. So you're going to want to put it in water baths of different temperatures. And I would always say a range, especially for SNAB, they like ranges. So I'd say 0 degrees to 30 degrees. You have to make it sensible because it is a plant. Well, it's not necessarily a plant, but if you put the temperature too high, like at 70, the enzymes are going to denature. We don't really need to do that, right? So 0 to 30, I think, is reasonable. Then control variable, volume of water, species of phytoplankton. I like doing, if I can, abiotic and biotic. Cool. So I've said my control variables. And now we know we need to get MPP and R. Cool. So I'm just thinking, okay, this is photosynthetic, right? Really, it's going to make its MPP through photosynthesis because it's not going to be eating anything. It's a primary producer. So we need to measure photosynthesis. How can we do that? Well, we're going to measure it based on the oxygen that it produces in the light. That will tell us the rate at which it's photosynthesizing. Cool. For respiration, we can then say, okay, what if we made it dark? If we make it dark, the oxygen produced, or the oxygen used up is going to be from respiration. So that's how you can measure, okay, so I've used the wrong word there, but it's going to be oxygen used. So if you measure the concentration when it's light, you can say, okay, that's it photosynthesizing. And then when it's dark, that's it respiring. And then you can calculate your respiration and your MPP, and then you can find out what the GPP is. Okay, so new context. So copepods is a group of zooplankton. They're small animals that feed on phytoplankton. In an experiment, they've looked at production of carbon dioxide and uptake of oxygen, measured at different temperatures, and you have the graph below. So just know this line of best fit wasn't here before. Now they said determine the mean ratio of carbon dioxide produced to oxygen consumed at 17 degrees. So I'm just thinking, okay, 17 degrees are my black dots. And I'm also just here thinking, okay, if I'm getting a mean ratio, surely I need to do a line of best fit and get a gradient. Cool. So that's what we're going to do. So I'm seeing here differences between that is 0.48. This is 0.55. So I'll just do 0.48 divided by 0.55, which is 0.87. So we've got 0.87. And I'm just thinking as well, they wanted a ratio, so we're going to say to 1. Cool. deduce two conclusions from the results. So I'm just thinking as well, okay, deduce means, let me pick something from the data and then write what that means. So in terms of what I can see in the data, it's very obvious that there's more O2 uptake at the lower temperature. There's also more carbon dioxide produced at a lower temperature. Cool. What does that mean? So we've said that, fine. That means that at the lower temperature, we're having more respiration. I should even mention at lower temp. So that means more respiration is going on. And that's one conclusion. I'm just thinking, what else? Well, if we're taking up oxygen, the respiration that we're doing is aerobic. So that's the other thing I'd mention. Okay, so here we have HIV, and it's a cause of acquired immune deficiency syndrome, or AIDS. So they've given us a diagram of HIV particle quite nicely done there. And the first question is, state the role of GP120 in the life cycle of HIV virus. So initially, all you're just thinking, okay, when HIV attacks a cell, normally the GP20 is going to bind to the CD4 in the T helper cell. And that's literally what you want to say. So it binds to CD4 in the T helper cell. The next question is, give a reason why there are proteins on the host cell in the lipid membrane surrounding the virus particle. So you can see here that the way they've drawn it, you've got the lipid membrane that's just out of it here. And you can see proteins on the host cell are there. So in terms of the reasoning for that, I would have said, okay, I'm still thinking about the HIV life cycle. Well, HIV particles undergo receptor-mediated endocytosis. So that means that when they bind, when the GP120 binds CD4, then the viral particle is then endocytosed, and in doing that, it takes from the host cell membrane. So I'd be saying along those points for this one. So I'm saying the virus gets the lipid membrane from the host cell. Essentially, that should then mean that the lipid membrane now has particles from the host cell. That's my kind of route to answering this question. Okay, so we get a bit more information saying that during viral replication, the viral mRNA is translated into a virus polyprotein called GAG. Cool. And then we're saying that GAG is split by HIV protease, and you have a diagram here. So the question is, deduce how HIV protease can split the GAG polyprotein into individual proteins. So when I see that we have a deduce question, I'm thinking, okay, I need to use information and use that to get to a conclusion. So what I'm looking at here, well, I can see really the information we have is that HIV protease is just acting in different places, so specific places. And that's what I'm going to say first. So the HIV protease binds specific places on the polyprotein. I think that would be fine. But what I also want to just add on, because we're talking about an enzyme here, complementary to the active site. I think that's just a good thing to add on in general. Then that's only going to be one of the marks. I need to think, how am I going to go over to? Well, I'm just going to elaborate on what happens once the polyprotein, well, the parts on the polyprotein are recognized. So I'm going to say hydrolysis of peptide bonds between adjacent amino acids. That should get me my other 2 marks. Okay, so it says HIV protease inhibitors are used in drugs to treat HIV infection. So some people, in some individuals, the HIV has resistance to these inhibitors, and they have a graph that shows the effect of polyprotein concentration on the initial rate of reaction of normal and inhibitor resistant. So the question is, calculate the initial rate of reaction in the inhibitor resistant HIV protease, right, for a polyprotein concentration of 7. Now, looking at our graph, we actually don't have anything for a polyprotein concentration of 7, so we know we need to extrapolate the data. So in order to do that, I need an equation for the line, and then I can kind of figure it out. So you should remember your y equals mx plus c, y equals mx plus c from GCSE maths. Now, okay, so what I'm going to do is I'm going to calculate the gradient of the inhibitor line. So I'm going to say that's about 2, and this is 5. So changing y over changing x, 2 divided by 5 is going to be 0.4. That's our gradient. Now, we can see that it's starting at the origin, so that must mean c is 0. So I'm happy to say y is equal to 0.4x. Yeah? Now, we want to say an x value is 7 because the polyprotein concentration is now 7. So I'll just do 0.4 times 7, which gets me 2.8. And I'll be happy to put that down as the answer. So just looking at units, 2.8 micromole per decimeter cubed per second. So 2.8 micromole. To be honest, I'm going to write the unit, but they almost never really care about units in A-level biology. So if you don't put the unit, don't worry. But just make sure you're getting the actual. Okay, and for the final part of the question, it says, explain the importance of enzyme and substrate concentrations when comparing the initial rates of reaction for different enzymes. So let's just tackle this one first. Enzyme concentration needs to be the same. So whenever you're doing a comparison between different enzymes or any like sort of practical procedure, that's your control variable, enzyme concentration, because we just want to compare the different enzymes themselves. Cool. Now, when we're talking about substrate concentration now, the second part, we need to think, okay, if we're investigating enzyme concentration, then the substrate needs to be in excess, or the substrate just needs to not be the limiting factor. Yeah? Because if it is, then that is going to inhibit how the enzymes can work. So that's something that you need to say. And essentially that's the next 2 points. So you're saying basically substrate concentration must be excess so that the enzymes can work at the optimal, right? Okay, so we have a bit of information here about MDMA causing damage to the bone. And to be honest, this is something that you should have learned from your lessons. So you should know MDMA inhibits the reuptake of serotonin. Yeah, that's something that you should have learned before. Now, that's what we first of all want to say. So we're explaining, so we need a biological principle as to why. But what you need to know is it's going to inhibit serotonin receptors on the presynaptic membrane, which means that you can't reuptake serotonin. Yeah, those are your first 2 points. Cool. If we have in the, so say this is presynaptic, this is postsynaptic, so what my bad drawing. What we're basically saying is serotonin is going to stay in the synaptic cleft. Yeah. So because serotonin is still there, it means that more serotonin can bind to the complementary receptors on the postsynaptic membrane. So when serotonin binds there, what it's going to do is it's going to trigger the opening of the sodium ion channels. And when the sodium ion channels open, you can imagine we're getting the influx of sodium, we reach the threshold potential, vortigated sodium ion channel opens, now we're getting an action potential. So that's the kind of thing that you want to be saying for this answer. You obviously don't need to go into so much detail about the actual process of an action potential, but definitely everything that's happening in that synapse, you definitely want to be talking. Okay, so we're doing a bit of a practical, and we're looking at MDMA, the behavior of mice. Cool. They've got an MDMA button and a control button. MDMA, they get a dose of MDMA, and with the control, they get some salt. So we've seen the results here, and our job is to determine the effect of MDMA on the number of button presses as well. When I'm initially looking at this data, we just need to think about what stands out to us. So we can clearly see, first of all, that if MDMA, they're pressing the button more than the control. Cool. I can also see from day 6, we see a big increase in the number of buttons with MDMA. And those are kind of the major findings, I would say. I think definitely I'm going to want to compare that this gap has gotten bigger as well. So those are the kind of considerations I'm thinking, because again, when we're saying determine, we need to compare it to what we had before as well. So that's kind of what we've kind of written here. So I said MDMA has increased the number of button presses. Cool. So after 6 days, the MDMA button presses is almost doubled. Yeah, it's literally almost doubled. And then I also wanted to do a calculation. So I said initially it was 8, now it's 16. So just thinking, what bits can I actually like pull out from the data that would be good things to say for the. Okay, so now they're saying MDMA can cause swelling of the brain, or edema, which causes death in some people. So we have a diagram here that's showing what's happening. So if they retain water, there's a fall in the blood solute concentration. So the brain swells, you constrict the blood vessels, die. Cool. It said that brain cells regulate the volume using active transport sodium ions. So first question is state what is meant by active transport. This one should be happy days. Very easy question in terms of if you know your definitions, you'll get this mark easy. So whenever you talk about active transport, and I think it's just worth learning this definition word for word, net movement of a molecule against the concentration gradient is important. I would add on food specific carrier proteins, but using ATP is the other really important bit here. That's what's mainly getting you the marks. But say the whole thing every single time, because you never know what the mark scheme is going to say. Cool. Now explain how active transport of sodium can regulate brain cell volume. So we need to think, okay, if we uptake sodium ions into our cells, what's going to happen? In terms of swelling and in terms of cell volume, they're trying to hint at osmosis. So what you're going to say is you're going to uptake sodium ions, which is going to cause an increase in solute concentration, or a reduction in water potential is also acceptable. If that's the case, then that means that water is going to move into the cell via osmosis down the water potential gradient. And then if we have more water in the cell, we're going to increase the hydrostatic pressure, which is why the cell volume is going to increase. Now, explain why swelling can result in vasoconstriction. So you just need to kind of think about this a bit more logically. Like, if the cells become swollen, they take up more space, yeah? And if they take up more space, they're going to compress the blood vessels, or that means that the blood vessels have less space. Cool. Now that these blood vessels don't have as much space, they're going to vasoconstrict because of the fact that they're going to be obstructed. So they need to vasoconstrict. Now, one more thing. You really need to be aware, be very aware that it's not the arteries that vasoconstrict. So if you say arteries, it's wrong. It's always arterioles. Arteries cannot vasoconstrict. It's only arterioles. Cool. Another thing that you might want to add on here that I haven't written down though is that when these blood vessels vasoconstrict, then you reduce the blood flow to the brain or in the brain, which is what's going to cause death, as we saw from our flow chart. So that's just an additional point that you can maybe add. But to me, I think the swelling and then compressing the available space or reducing the space for blood vessels should be enough. Okay, and this is a question that comes up essentially every year in a different format, but you are going to have to say the same thing pretty much. So whenever we're talking about death of cells, normally what you're saying is that there's a reduction in blood flow. So in this case, it's because of the vasoconstriction that you're reducing the blood flow. And then if we have a reduced blood flow, always say that means that we have less nutrients or oxygen, normally oxygen. We have less oxygen going to the cells for respiration. Like whenever you're talking about death of cells, always relate it to lack of oxygen or lack of nutrients and lack of respiration. That will normally get you the mark. So that's what you'd say for that. Okay, so again, this is the 9-mark question. I'll just say it straight off the bat. So we're not going to write out an answer, but we are going to talk in detail about what kind of considerations you need to have when answering the question. So let's just read the context. This is going to be so important when we're answering the question. So sports injuries can be treated to reduce the pain caused by muscle cramp. Cool. Ice massage, ice pack, and cold water immersion are 3 different types of treatment. The compound action potential is a measure of the sum of all action potentials in a muscle. In one investigation, the effect of treatments on the compounded action potential in a leg muscle is investigated. And now they're saying for one action potential, they want to talk about latency, duration, amplitude. Cool. So what I'm thinking here is I need to kind of relate these terms, and they've given some definitions. So latency is the time taken for application of a similar sort of the CAP. Duration is the length of time with the CAP, shows depolarization. Amplitude is the sum of the depolarizations, and conduction velocity is the speed. Okay. So I'm just thinking, how can I link this to biological concepts I already know? Yeah, and this is without seeing the question first. So I'm thinking latency time. If I draw out action potential graph here, latency would be describing this bit here where sodium ions are coming into the cell. Okay, I'm going to talk about that in my answer at some point. Duration, the length of time that it shows an action potential. Okay, I'm thinking that's going to be the time when all the sodium ions are flowing in. So that's kind of describing this bit. So again, I might be saying that in my answer, yeah, in terms of duration. When we're talking about amplitude is more, to me, if I'm talking sum of depolarizations of all the individual action potentials, I'm thinking that's kind of a point about summation potentially. So if I spelled it wrong, but I'm thinking that might be about summation. So I'm going to mention that summation. There we go. So I'm going to maybe talk about summation at some point. And the conduction velocity, we're aware, is just the speed of the action potentials. So now that we've kind of done that, we can look at the data here. So it says the effects of different treatments on CAP for motor and sensory neurons are shown in the graph. In black, we have pre-treatment. In white, we have post-treatment. So before we look at that, let's actually look at the question. We're saying assess the effect of core treatments on nerve conduction and reduction of pain causing muscle cramps. Use all the information provided in the question as well as your own knowledge. Cool. So when we say all the information provided, we're looking at all the parts of the question. So in terms of the initial information, I'm thinking, okay, reduction of pain is going to be sensory and muscle cramp is going to be motor. So if we lower motor impulses, we get less muscle cramp. If we lower sensory, we get less pain. Cool. That's what we're kind of thinking. And we've defined the term so we know what to talk about. Now, looking at the first graph here, latency. So we talked about latency being the time it takes to prepare. So from sodium ions coming in to reach the threshold. Yeah. Let's see what's happened here. Well, ice massage, ice pack, they're roughly the same for motor, but you can see in the cold water we've really increased that time. So that's something we can mention. In the sensory neuron, you can see all of them have quite a decent increase. Most in the cold water, I might mention that, but all of them have an increase. So we're saying that latency is going to be longer. So it's going to take longer to actually generate an action potential in all of them. That's good. Because that, if it takes longer, then that means that we get less cramp with cold water. Maybe not so much for ice pack and ice massage, but less cramp in the cold water for sure. And then less pain for all of them, but mostly in the cold water. Okay, let's look at that's the duration. So when we're thinking about the duration, what we said is, okay, duration is the time that the action potential is going on, as in you've got the voltage gated sodium channels open. So what I can see here is, in terms of duration, similar for ice pack, ice massage, but again, cold water is increased a lot for the motor neuron. So if we have more, then that means our muscles are contracted for longer. Yeah, because it's the motor neuron, so it's going to cause the contraction of the muscle. Whereas here, again, we just have it longer for the cold. So in terms of that, that means the action potential, again, is going longer in the sensory neuron. Now, if the duration of the action potential is longer, of course it means that we might have it contracted for longer. Yes, okay. But when we think about cramp, is that a good thing? I'd say yes. Because if it's contracted for longer, cramp is kind of caused by the involuntary contraction and relaxation. So if we have it in one state, it's actually probably better for us. Also, if the duration is longer, then that means that we don't repolarize because we're still in this place. You can see it. We're still in this place. So because it's not going to be able to repolarize, we actually aren't going to get enough action potential for a while, which means that the pain signals are going to be slower. Cool. Amplitude, we talked about maybe being about summation. So amplitude being like how we're getting more and more action potentials per se. So in terms of that, what I can see is very similar for ice pack, ice massage. Cold water has gone down a bit for the motor neuron. And then you can see here for the sensory neuron, well, ice massage and ice pack, see it's gone down quite a lot. But it's actually gone up in the cold water, which is interesting. Now, what is quite good about that is if we're lowering the amplitude, we get fewer impulses, which again is going to mean we're going to get less cramp, we're going to get less pain for the most part. Yeah, but I definitely mentioned this. This seems to be a bit of something that is interesting. And then here again, when we said conduction velocity, again, we're just talking about the speed. So again, quite similar for ice pack, ice massage, but cold water, it's gone down quite a lot. And then it's gone down quite a lot in all the sensory ones as well, which means we're going to get less of the pain signals. So we've taken our time to go through all the data, and hopefully through that, we've also linked back to the kind of physiological aspects which are going on in terms of the movement of the sodium ions and everything. So when we've now got all the data, remember, prompt was assess. So we've looked at all the data, we've talked about it. Now we need to come to making conclusions. Cool. What we're saying is, okay, well, to be honest, for all of the treatments, especially in sensory neurons, they were quite good at doing their job. You can maybe mention duration, maybe not so much, but for the most part, they really did their job. So I'm going to say most of the treatments or all of the treatments are likely to reduce pain because we have reduced sensory nerve conduction. Cool. What we're also going to say, okay, in terms of the motor ones, I'm not going to lie to you. Well, it seemed like ice pack and ice massage weren't that great for motor, but cold water really was. So if cold water is really good at reducing motor signals and it also reduces sensory signals, what we can definitely say is it's going to reduce cramping and pain. So maybe this would tell you that maybe cold water is the best treatment. Cool. Now it's a 9 marker. So what I also really like to do is just kind of critique our thing a bit. So in terms of the limitations of the study, let's quickly see what was done. So they said ice massage, ice pack, and cold water immersion. One thing they haven't mentioned is I don't know anything about the temperature used. So maybe the cold water was genuinely just colder than the ice massage and the ice pack. I don't know. So temperature is something that I'd probably mention here. They've said a leg muscle. Okay, you'd assume it's from a human, but they've seemed to have only used one leg. So if they only use a leg from one person, then that could obviously affect the values. We don't know if they've done repeats, blah, blah, blah. That's something that could be at play here. What I'd also mention as well is, just looking at the data, I don't see any error bars. So error bars would go a long way to tell us, is the result significant, is it not significant? That's another thing. There's literally kind of no information whether they've done repeats and made sure that this is valid. So I definitely want to be mentioning those as well at the end, just to make sure that I've kind of critiqued it a bit, and that should help me to kind of elevate my answer. Okay, we're on the article now, and having had a brief read through, it seems like a lot of it was about evolution. So they said that explain why analysis of amino acid sequence in this posing can be used to determine phylogenetic relationship with different sloths. Now, to be honest, I don't really think you need to read the article to get that question, but let's just read box one. So having read through it, we're talking about sloths and some different armadillos and anteaters in this order. they said that analysis of the amino acid sequence. So we're talking specifically about the amino acid sequence, and we should know, based on our knowledge of A-level biology, that when you talk about different sequences of amino acids, you can use that to classify organisms, such as you can use different sequence of DNA. And we're talking about the specific islands protein, and we're saying it's confirmed that these. So actually, to be honest, from that you can infer that also the armadillos and the anteaters also have these proteins. And it's saying that that meant that they could class them into something different. So they want a new classification for the sloths to put them into different families. Okay. Yeah. So in terms of now answering this question that we had, what we can then say is, to be honest, I didn't include this because I didn't read it that closely before, but we can say the islands protein is actually present in all of the sloths, or it's present in all of that order. Yeah, cool. Once you've said that, we know that the amino acid sequence is determined by the gene. Yeah, so that's what we can say. I've said it's determined by the sequence of bases in DNA, but it's determined by the gene. Cool. Now, let's get into what's happened. So, with the phylogenetic relationship between different sloths, what we know is if there's different sloths, there's been a mutation at some point. So I'm going to say mutations occurred. That's caused a change in the DNA base sequence, which changes the body sequence of codons and mRNA, which now changes the amino acid sequence. Yeah. If that's happened, the differences in the amino acid sequence we can use to classify into different groups, or just in relation to the question, we can split them into different branches in the phylogenetic. Okay, so let's describe how algae living in the hair of sloths can be shown to belong to one of four phyla. So again, to be honest, I don't really need to read box 2 to be able to answer that question. I think you can kind of tell it's going to be talking about sequencing of some kind, or maybe even PCR gel electrophoresis. So when I'm saying, okay, algae have been cultured, again, we're thinking they've done some sort of analysis of maybe a protein or a gene. That's all you really need to know. So there's kind of two ways you can answer this. I've gone down DNA gel electrophoresis. So in that instance, again, we don't really need to use the actual article. You just use your own knowledge. So just describe what's happening in electrophoresis in terms of you get the sample, amplify it, restriction on the nucleases, and then you analyze it using electrophoresis and compare the binding patterns to the phyla, because you always have to make sure that you're relating back to the question. Another way you could do this is just say, okay, you took a gene. To be honest, in that case, maybe then we could go back to the box and pick out an example of something that they mentioned. Because then we're just going to say analyze and determine some similarities, and then you should be able to get your marks. So again, it's nice to use the article for context. However, that's another question that's more based. Okay, so it says describe how spirometry can be used to estimate the metabolic rate of a sloth. And again, although you could argue, again, we're just using our knowledge. Let's just do due diligence and look at here. So in terms of the information here, it's talking a lot about the metabolism. And then all I can see is kilojoules per day per kilogram. So if we're using kilojoules per day per kilogram, I'm thinking that's kind of the units that I'm going to have to use as well, won't I? So that's what I've got from the article. Now I'm thinking, okay, in terms of a spirometry practical, what we want to do is our control variables, same temperature. We also want to talk about use of soda lime so that we can absorb the carbon dioxide. So that, you know, when you're having the change in the volume, it's because the oxygen is being consumed. Cool. Now, in terms of the spirometry trace, your trace should look something like that. In order to get the oxygen consumption, say that on bad drawing, you need to do from the height of the peaks, and you need to just calculate that change over time. So whatever the change in volume is, divided by time, is going to give you the rate of oxygen consumption. Yeah, that's the kind of thing that you want to describe. Yeah, cool. We also saw that it was in kilojoules per day per kilogram. So what we're going to want to do is we need to get the mass of the sloth so we can do this kilogram part. And we also need to convert oxygen to kilojoules. And then we can also convert the time to the day as well. That will give us the kind of thing that we're looking for in that question. Now, to explain how the presence of different antibodies can be used to show that some viruses are specific to sloths and some are incidental. So I just want to get some context as to what specific and incidental mean pretty much. So if I read box 3, let's see. They said that amphipod-borne viruses. Okay. And I guess essential means specific to it. So you can see here, actually, this is important. So, so if I got a bit quiet, it's because I'm reading. But it says characterized according to antibody and virus isolation data as a sloth being specific. Cool. So that gives us enough information to go now and answer the question. So when I'm thinking what we're going to say now, I'm thinking, okay, well, virus antibodies, what's really the case here? Well, antigens are specific to the virus, and then the antibodies are complementary to the virus's antigens. So those are the first two things that I'm going to say. Cool. Once I've said that, okay, if it's specific, it means that this virus is only found in sloths. So the antibodies produced will only be in sloths. That's what the article's kind of alluded to. If it's incidental, then that means that it's found in other organisms. So if we analyze and find that the antibodies are also present in different organisms, then it's only incidental. And that's how we're gonna answer. Okay, so sloths have the longest digestive process on record for a plant-eating animal, paragraph 7. So again, I'm just gonna quickly read for the context, especially because it was a deuce question. So it says that they nibble a bit here and there. Cool. They don't rush digestion. They take a really long time to digest swallowed food. Plant eating mammal. So it's underline it properly. Plant eating mammal as well. Okay, interesting. And also just a hint here for the next question. It says this one big animals take longer to digest their food would just mean that they're saying sloths are small. Okay. So if we go back to this question, one thing is they said plant-eating mammal, right? Plants have a cell wall making them notoriously difficult to eat. So that's the first thing we're gonna say. I think when you get an article and you realize that they're talking a lot about sloths, I would just before the exam do a bit of research into like sloths, for example, and then that's how you'd be able to know, okay, these are small animals. Cool. If that digestive process is taking so long, again, you're going to need to think about digestion, what's kind of happening in digestion. And I appreciate they don't cover a lot in SNAB, but what you should know is those digestive enzymes are going to be working. Yeah, we've kind of touched on this maybe a bit like module 2 cystic fibrosis in terms of digestive enzymes. Yeah, that's going to allow them to do their function to get your nutrients. Yeah, that's the kind of thing you want to say. Now, paragraph 8 and 9, it said maintaining a core body temperature is energetically expensive for the sloth. So again, we're going to go to paragraph 8 and 9 and have a bit of a read. So it says, in terms of conserving energy, and again, you would have been expected to maybe do a bit of research on how sloths conserve energy before the exam. But body temperature varies widely. We know they're small. Think about how that relates to the body temperature. They said that swings of nearly 5 degrees is crazy as the environment is changing around them. So that's something. And they've said it's because maintaining the core body temperature is energetically expensive. I was thinking, okay, how are you going to maintain a core body temperature? It's going to have to do with homeostasis. So we're going to have to talk about that eventually. Now, they said that they need to warm up. Shivering is done by a lot of animals with energy to burn. So we're kind of hinting that the sloths don't have that. But what we're saying is the sloths are instead going to climb higher into the canopy each morning, right? So I think that's given us enough context to maybe go for the question. So what I would say, okay, sloths are small animals. So they have a large surface area to volume ratio. That's why the environment can change the temperature so much. What we were saying is that the temperature changes a lot. So like that's why they go to the upper canopy when they want to warm up because it's closer to the sun. So you can mention something about that as well. That will help them to save energy. Yeah, that will help them to save energy as well. And then we also saw, okay, they don't really undergo homeostasis. That's what the article was kind of saying. So we're saying homeostatic mechanisms, such as shivering, for example, something that was mentioned, requires a lot of energy. And then finally here, they said sloths can warm up by climbing high up into the canopy. To be honest, I don't really need to go back to the article to read this. That's just behavioral. So you know you have different types of adaptations, but this one is very much just okay. So they're talking about sloths hang upside down, constant grip possible by a lattice of tendons. So that's in paragraph 10. I'm just going to quickly read paragraph 10 to see if I can get any more context before giving you that question. So they said slow walking, camouflage. So main predators are not seeing the prey move. Maybe that's relevant. They said hanging upside down. Oh my goodness. Hanging upside down, completely still for hours. So completely still for hours, okay. Of course, and then that's the bit that we have, don't we? Constant made possible. Okay. So I think what you're kind of getting at here is that they're using tendons more than muscles, and they're like saying that the idea about constant grip, so not really tiring out compared to other organisms, and I think that really does give you a hint as to what you want to say. So really, what we're saying is, okay, you're using tendons instead of muscles, essentially. So when I'm reading that, if I had that before, I would just maybe do a bit of research into like what tendons kind of are, pretty much. But they kind of attach muscle to bone, essentially. So what I really want to say is that like your tendons don't really contract and relax like muscles. So that's one difference between the two that I can put down. What I'm also going to think about, okay, well, if we have a lattice of tendons, that means that we don't really need muscles to grip. Yeah, that's one thing. They don't really need the muscles to grip. So what I'm also going to think about, okay, why is it a benefit really using tendons instead of muscles? Well, we know that muscles need energy to contract. Yeah, tendons don't. So that means that we don't need to use energy to maintain the grip, which is why they can stay so still for so long. And also your tendons aren't going to fatigue. So because the tendons don't fatigue, unlike the muscles, that's why they can stay so still for long and they don't get predated by the owls. Yeah? Okay. They said sloth muscles contain a unique set of enzymes. So we're going to go to paragraph 12. So looking at paragraph 12, let's see. I said the butchers dissected sloth cadavers. They said have very little muscle, which is what we mentioned before. It's just not really relevant to the question that we had. So they said most strikingly, there's a unique set of enzymes in the sloth muscle. That's what we said. Tolerance to heavy accumulations of lactic acid. Interesting. Okay. So if we go back to our question now, it's something to do. So we need to get some information from the text and then say, why do sloth muscles differ from most human muscles? Yeah, and it's going to be related to the enzymes, as we said. So what I really say is that, okay, first of all, let's go back to basics, biological knowledge. We know that when you have difference in enzymes, always go back to difference because of amino acids, german peptide bonds, which means your R groups are in different positions, which means bonding between R groups with two named examples are different. Yeah, that's the first thing I want to say. Cool. Now, I found the point that I talked about lactic acid very interesting. So I definitely want to just mention that. So what I would say is that they're more resistant to denaturation. I don't know why I said temperature, lower pH. That's what I would say at lower pH because lactic acid is going to reduce your pH. So I just maybe mention that as well when I'm answering. Okay, so by the looks of it, we need to go to box 4 as the final bit of this. So reading box 4, sloths, they have a lot of arthropods, but like the flies and mosquitoes, and this is what we had. There's a specific species of beetles, mites, and moths. They said the sloth moths get protection from predators. If I can highlight that, protection from predators, nutrients in the sloth skin. And then this other bit, the sympatry. So sympatric speciation. Okay, interesting. Okay, so in terms of community, what we just need to do is just define community. So definition we should know: population and species occupy the same habitat. Then I was thinking, why is it a 2-mark question, really? To be honest, so I would just then also just include okay, they benefit from living on the sloth, for example, by being protected by predators. That's what they said in the question. So that's what I would kind of say. That other point, this added point, I think is a bit mean, but either way. And then sympatric speciation. Again, to be honest, this one you don't really need the article for. You literally just need to know what sympatric speciation is. So in terms of sympatric speciation, that's when you have speciation like within the same area. So allopatric is when they're separated. Sympatric, they're living in the same area. So there needs to be a reason why they're reproductively isolated. So that might be, I think the easiest one is normally just different genitalia. You could obviously have different feeding times or mating periods or anything like that. But I've just gone different genitalia. So that means that they're reproductively isolated, which prevents gene flow between the populations. So they develop different gene pools. That's just your standard answer for sympatric speciation. But that wraps up this.
Okay, so that is a wrap on the paper. I hope it was really useful in terms of going through the questions, getting a bit of insight into how to approach them and how to kind of read the context clues, especially in a question like this, which is very, very context heavy. So, hope it's been useful. If it was, please like the video and subscribe to the channel. Any questions at all about SNAB biology, biology in general, or paper 3, put them in the comments below and I'll do my best to answer. We've also got our TikTok and our Instagram, both at A-Demola. However, that's all for me for now. Good luck for your exams and we'll see you in the next one. Bye for now.